Great. Thanks everyone for sticking it out to the end here. I hope to make it worth your time. I'm Michael Chu, the CEO of Kytopen, and I'm excited to tell you about our FlowFect non-viral transfection technology. This is an improvement over existing non-viral, mechanical, electroporation, chemical technologies, and it'll help move the industry faster towards clinic and bring in these non-viral methods. So we're at a cell and gene therapy conference. We all kind of know the potential of these therapies, and we know how much is invested in them. We also know that the actual reality is we've probably brought, I think, 20,000 patients to date, brought these therapies to those patients. One of the limitations, one of the things that is preventing us from moving faster is viral transduction methods, the safety and cost of these methods. And so there's a big push to look at non-viral methods for doing the cell engineering, and that's where we focused our time. So Kytopen provides fast, non-viral tools for cell engineering. And they work across a broad array of cell types and payloads. Our FlowFect platform provides continuous electrochemical mechanical transfection that can operate at any scale. So you can see, I think the video only ran once, but you can see down the lower right-hand side sort of a scale view of our flow cell. It's essentially a metal tube interrupted by a restrictor. That restrictor provides some mechanical disruption of the cells. The metal tube is our electrodes for electroporation. Cells and payload are suspended in a proprietary buffer. And as the cells and payload flow through the center of the flow cell, if the video were running, you'd see we can arbitrarily vary the frequency, waveform, power of the electric field. And so that flexibility and inclusivity of mechanical, electrical, and chemical gives us a very wide operating range to work across cell types and payloads. There we go. So the FlowFect platform is built on scalability, and the fact that it's continuous flow rather than a fixed batch gives it several compelling advantages over other non-viral methods. And first is that you can run at very small volumes. We have the ability to run sort of at the microliter scale, and that enables sort of high throughput screening and for process development. So you'll see later we have a tool that can run a 96-well plate in about 15 minutes at about 100 microliters. So we do have a broader operating range. We have ways of figuring out the optimal operating point across those parameters. At clinical volumes, because it's again a flow-based rather than a batch-based, if you need to get to higher volumes, you can use the exact same flow cell and just run it for longer essentially and produce clinical volumes. And when you're ready to get to GMP manufacture, again, same flow cell, same parameters developed at PD. Essentially, you can just run for longer time for higher volume. So this essentially lets you run your discovery and PD processes much faster than you normally could across a larger range of variables, and at the same time, once you found that optimal, you can translate to clinic and manufacture sort of seamlessly without having to redo at scale up. I may have gone, did I jump ahead? No. So the result is a simple, easy to use and powerful platform for cell engineering and for cell manufacture at scale. And it works across a wide range of cells. We have applications in cell therapy, CAR T, CAR NK, sort of the typical sweet spot in that field. There are applications in gene therapy, also loading extracellular vesicles, any sort of gene modification there. Cell line development, again, anytime you do cell engineering, if you need to put payloads inside of cells for protein production or other cell modification, the high throughput screening aspect of the transfection tool is very powerful. And finally, all of these processes require, optimization of payloads, media, and whatnot, and the tool there is also used to get those optimized supporting chemicals. Yeah, let's move on. So what does this look like? We offer sort of a range of products and services. On the left is our FlowFect Discover platform. It's a high throughput screening robot that has the flow cell integrated in. You'll see a video of it a little bit later. It can run a 96 well plate in about 15 minutes at volumes of 50 to 100 microliters. Once the optimal parameters are established, there's a benchtop unit, the FlowFect TX, so closed, sterile system, can run at larger volumes, either kind of syringe to syringe or bag to bag, and can scale up to liter scale and greater depending on the time and the parallelism that you run at. There's a developing version for GMP as well, so when you get to manufacture, we'll be ready for that. We have a software platform, FlowFect Connect, that allows you to sort of arbitrarily work with any and all the variables involved in your transfection development. You can vary the electrical properties, the flow rates, and so on. It's your recipe. We'll help you develop it, but it's a sort of totally open book, right? So you can have as much or as little variability as you like. And finally, we offer services to help you both learn how to use the machine and run the machine. If it's not your primary interest in doing the optimization, we can do that for you. We can run complex DOEs and statistical analysis to help you get to the operating point that suits your needs. So we often find that customers they get their IND and they want to get to clinic as fast as possible. Often it's hard to justify doing process development when clinical results are the most important thing they need to hit their next milestone, the next funding round, and so on. One of the big benefits that we have is speed in process development. You can see here there's a case study for process optimization. This is I think it's a CAR-NK cell with a knock-in, and CRISPR knockout using primary cells. This 9-week experiment ran 4 plates, 324 experiments, about less than 100K to run the entire panel. And we were able to vary, across, take tests across multiple donors, multiple cell and payload concentrations, multiple gene targets, and then vary the transfection parameters themselves, the EP energy, the flow rate, the time in process, and so on. And I create sort of a robust data set that allows us to find the point that has sort of the highest efficiency, the highest cell viability, and the least amount of energy put into the cells, so they have the fastest recovery time. So this was 9 weeks. Typically, this process might take 12 to 18 months. It's short enough that you could probably run this in parallel with your clinical development work and get the optimization PD done before you go to clinic, so that you're not locked into a non-optimal process when you go from clinic to manufacture. A bit of data on the performance. We can use this across a range of cells, including T cells, CD34 positive, B cell, NK, and whatnot. The top slides show mRNA delivery at high viability, post-transfection and versus a control. With no transfection, you can see upper left the viability numbers across 3 different cell types and against a control. So very little loss in viability, and then you can also see the efficiency versus control on the right. Lower graphs show CRISPR-RNP knockout across 3 different cell types. Looks like the colors didn't come through on the slide, but the left-hand columns are the Kytopen results. The right-hand is a static EP, sort of an optimized process from a customer. We came in, ran our optimization, and we were basically able to reproduce the results in a short amount of time. And the results 48 hours after transfection, showed durability in the knockout. So the other aspect of the benefit of the Kytopen platform is speed to transfer to manufacturing. Because we're this flow process, again, going to higher volumes is really just running the system for more time. You can see here an example where we did discovery at 100 microliters, and then we ran the exact same process parameters at scale at 10 milliliters. And resulted in substantially the same viability and efficiency. The only difference between these 2 data sets is that the left-hand column was run for 120 microseconds. The right-hand column was run for 12 seconds. We produced order of magnitude, or several orders of magnitude more transfected cells. So you can imagine, for getting through clinic, you can run a clinical scale quite easily for allogeneic therapies. If you need to get to higher volume post-clinic, it's a fairly straightforward path to doing that. And we've demonstrated volumes over a liter. I think easily get to 5, and you could run systems in parallel to get to higher volumes. So some quick thoughts on comparisons and the value proposition here. It starts with the power of the Discover system to run rapid process development and accelerate your PD through screening and small volumes. This shortens your development time and allows you to better optimize your processes. Optimization can include lower EP energy, which I think is a big impact on recovery time and viability, and result in sort of a better process overall. The next value is a rapid scale up. As I showed, going from discovery to manufacture, the PD effort is very minimal, and continuous flow lets you get to large volumes, essentially unlimited process volumes as you get to scale. And finally, I didn't really touch on this in the data, but because of the range of operating parameters we have and the ability to optimize them, we can deliver not just into the cell but into the nucleus. We have some data that shows that, and that makes it compelling for therapies that require that. So a couple pictures of the system kind of in the wild. I'll look at the video right now. First, this is the fluid handling robot. It just picked up cells 8 at a time. Now it's picking up the buffer and payloads. The flow cell is at the tip of the pipette, and so they draw the material through with no electrical energy. It goes into that black box, which is where the flow transfection happens, and we apply the electrical current in that maybe 5-second period when it's in the box. We just did 8 transfections. It gets rid of the tips, picks up new ones, and repeats the cycle. Total process time for a 96 well plate then is, it's less than a minute per cycle, less than 15 minutes for the entire process. You can see on the left-hand picture, both the Discover system on a bench and then To the left of that, with a white panel and the screen, is our benchtop system. There's a closed, sterile flow cell with weldable connections that bags hang on either side. The flow device sits in the receptacle in the center, and tubing goes through the pump. Simple user interface, GMP compliant software, and you can run production on that at clinical and manufacturing scale. We have a number of these systems out in the field, both at academic medical centers, CDMOs, and a few pharma companies as well. I can't list them all, but they're being used in anger at this point, and we're getting some great results. So how can you work with us? We're excited to engage new customers on a commercial basis. You know, if you have applications that fit within our current set of demonstrated capability, we'd love to talk to you and help you accelerate your product development. You know, we can work on your manufacturing challenges. We can leverage our existing tools and processes. If you have an application that you're struggling with that is new to us, we'd also like to work with you. I think we have the broadest range of capability to solve these transfection problems. We'd love to collaborate and figure out new applications for the technology. And finally, the FlowFect flow cell is imminently integratable, and I think it makes a lot of sense to be thinking up and downstream and where this can fit in a fully integrated process, and we're looking for partners to explore how that might work as well. Finally, we're on a fundraising, we're launching a fundraising round soon, and to get some growth capital to bring this technology to a wider audience, build out applications, and so if that's your thing, I'd love to talk to you after the session. That's it.